pora branching off after 𝗗𝒄𝒄𝒏, it is actually sister to multipora. Okay, so overall, as I mentioned, typically families are quite well supported. So basically, initially we know that 𝗗𝒄𝒄𝒏 is incorporated. 𝒄𝒄𝒏 as a whole is well supported by genomics as well. It's just that certain genera may not, the relationships between genera may not be strong, right? So in this case, genomics, in this case, transcriptomes have shown that 𝗗𝒄𝒄𝒏 is very well supported as a sister group to multipora. Right, so this is what we can expect going forward as well for other groups. Okay, so taking a step back, right? So we've seen how certain in 𝗗𝒄𝒄𝒏 at least you have the gene trees and genomics more or less being consistent. And we also see that actually at the broad phylogenetic level, those clades which were well supported with gene trees are also well supported with genomic trees, in this case transcriptomes, right? So families are in the black and white alternating coloration. You can see that they we have known these families since we have sequenced them, right? So they are well supported. Okay, but where they were very unstable with single genes, right? They are now being unstable because of the genomic data. So one of these very major clades that we talked about was the basal clade. Remember, the basal clade was the first branching group around 450 million years ago, and then you have the complex and the robust clades. Now, if you look at this tree, this is a mitogenome tree that was built by Kidahara's group just a few years ago, shows that the basal clade is not a clade anymore, right? Because we have Microbacillidae. Again, these are deep water corals, so maybe we are not familiar with this that much. They have Microbacillidae, you have Gardneridae, and they are not forming a clade, right? But it's pretty weak still. So mitogenomes, 13 mitochondrial genes show that, again, this is not a good group. And following that, we have the hybrid capture tree by Andrew Quattrini. If you zoom into the 𝗖𝒄𝒄𝒏, this is 𝗖𝒄𝒄𝒏, you can see that there is no longer any basal clade. In fact, the basal clade here is, this is Microbacillidae. It is not sister to all of 𝗖𝒄𝒄𝒏 anymore, it's sister to the rest of the robust. Right? So that shows us that if a group, right, at the family level was weak in the first place with gene trees, they may shift around with genome trees, with genome data. All right. So when we did the same similar analysis with expanded group of expanded sampling across the entire 𝗖𝒄𝒄𝒏, we also see the same thing. So basically, only complex and robust, no longer any basal. So this is robust clade. You can see the robust is now including the basal clade now, right? There is a single family, Gardneridae, which hasn't been sampled before. So the jury is still out, but we think that there is no longer going to be a basal clade. So it's just robust and complex. All right. I was going to mention, all right, so going back to the complex clade, you can see that where they were supported by gene trees, so all these families were supported by gene trees, they are still well supported. So those without any values at the nodes, these are all 100% support for them, right? Because we have hundreds of loci, and so it's showing that these are very well supported. Okay, 𝗗𝒄𝒏 is a kind of a problem, and I'll get to that maybe later on, right? But robust clade can be fairly certain as well that those families are good families, right? Yeah, of course, there are some weird ones like Echinophilidae, but generally, these problems are restricted to the deep sea clades, so those azosynthetic groups. And that, again, that is a taxonomic bias that we need to resolve as well. Okay. So there's another set of analysis that were done. This is called the red-seq, and so this is basically using restriction enzymes to cut the genomes at specific points so that they can be the homologous genes can be compared, but this is restricted to fairly small groups. So you have parietes or at the genus level. This is fairly effective in distinguishing species. So this has been done for instance, for parietes. uh, uh, and you can see that what we expect changes will happen is in how we classify varieties. There hasn't been any revision so far, at least not at a large scale. But you can see quite clearly that there are some groups that sort of make sense now. A lot of the massive species, right, Lutea, lobata, solida, and so on, these tend to be massive, but they were split into different morphotypes or ecotypes for various reasons. They tend to be one single lineage, right? So clade number 5 is this one. Appears to be one single species. But again, the jury is still out. Maybe 𝗏𝗔𝒄 doesn't give us the right resolution. Hopefully, we'll get some answers in the next 3 or 4 years. But consensus seems, across a few studies, seems to show that varieties diversity is a bit lower than expected, right? So this is one way in which we are not splitting species. We may actually be combining or lumping species with genomic data. But, you know, as ecologists, if we identify ecotypes, that's really important for local identification. So perhaps the global taxonomic revisions is not going to affect how you identify species. You might just lump them eventually if you think that taxonomy is an important part of your study, but calling them different ecotypes is equally important as well. Pocilloporia, there have been a lot of work because Pocilloporia again is very widespread. Different species have a lot of plastic characters. In fact, is one of those poster child of Veron's plasticity across on the reef. So you have 𝗏𝗏𝗂𝗏𝒂𝙩𝒊𝙘 that looks completely different in various parts of the reef. It turns out that actually the morphology is a fairly good gauge of what the species should be. So those thick, clumpy ones, you know, 𝗏𝗏𝗂𝗏𝒂𝙗𝒃, acuta are a bit more branchy. But again, there are exceptions, right? But what is a good thing here is showing that 𝗏𝗔𝒄 as a whole is supporting the mitochondrial genotypes, right, that people have been using, right, for the last decade or so. There's this marker that you sequence the control region, and that can tell you what species it is, 𝗏𝗏𝗂𝗏𝒃, acuta, varicose, and so on. That appears to hold up at the genomic level, right? So we can have, this is one good example where if you have the mitochondrial gene sequence with good skeletal reference and a photo, eventually you might get to a point where you can just use the photos for identification. Yeah, but again, many of these groups, many of these species have been well known, the differences, so they tend to hold up. Okay, Leptestria, again, quite consistent with what we know, right? So using 𝗏𝗔𝒄 again, showing that there are some fairly widespread species, transversa, purpurea, but they are also very restricted species. So we have 𝗏𝗏𝗏𝗏, inequalis, but again, the sampling is not complete, so it might show certain species to be more widespread. But overall, what this is showing that is, in this case, is that the morphology have relatively been consistent with the genes, the gene trees, as well as genomics. One exception is this group called Leptestria purpurea and pruinosa, which I think a lot of us have issues trying to distinguish them. Often we say pruinosa is the one that, you know, the polyps are out in the day, and then purpurea is not, right? So, and amongst other characteristics, but this is showing that actually they might be the same species, right? The formal revision has been done, so this is one of those clear results which allowed the authors, in this case Roberto Arrigoni and Francesca Benzoni, to merge these species or synonymize pruinosa and the purpurea. Okay, so not much change, a lot of new species again, but not a lot of changes at the species level. Okay, we come to corpora, and that's where I think people will find that this is where the frustration will start, because corpora in some ways is one of the more difficult groups to identify because there are so many species. And I think the groups like Andrew Baird, Peter Kalman, Tom Bridgday, and Zoe Richards, they have put together genomic data sets that show that the relationships are actually contrary to other groups. They are not congruent with the morphogroups, right? But this has been known for a long time, right? For over a decade, right? Zoe Richards have already used gene trees to show that actually those groups, those species groups that we have been using have not been very effective, right? To distinguish them at the evolutionary level. But again, in this case, this is one group where most Acropora have the same sensitivity to climate change, to global warming. Maybe that's not a big problem. But if you are interested in identifying them to species, then I think you may have to not use those species groups. Or if you use species groups, we have to be cognizant that they don't form natural groups or clades, right? So for instance, you have some of these species groups, each of these letters represent a species group, and these have been put together, but you can still see that they are not, things that are called the same thing, in the same, groups. They are over the Robusta group is all over the tree, right? So this is based on the hybrid capture method that we've been talking about. We'll talk a bit more about Acropora later on. But moving on to just a local, that's why I think it's important to emphasize again that local identification actually still works, because in Japan, at least in Okinawa, there are 3 of these table corals, Hyacinthus, Cytherea, and Bifurcata, very well demarcated morphologically, right? And very well demarcated genetically and genomically as well. So if you are able to figure out what the species are at the local level, they tend to be potentially well distinguished by genes as well, and also by genomics, right? It is clear also that some groups like this one is not as congruent with mitochondrial DNA, but certain nuclear genes actually do distinguish them quite well. So this is the mitochondrial tree, doesn't distinguish them well, but you have genomics that distinguish them quite well, as well as morphology, right? So they are clearly distinct genetically, genomically, and as well as their morphology. Okay, so local identification is still very good. At the broader level, for instance, for the fungi, I think this is a problem that Nippon mentioned. You have groups like Lithophyllum, which comprises both solitary and colonial species. This is a big problem, and I think this will continue to be a group that will be revised over time, because groups like this study have shown that there is a lot of cryptic species, right? Even for Hippolyta limax, right? So this is one species that we all identify quite well across the entire Southeast Asia. Turns out there are multiple lineages. There is no revisions yet for this. I think it's not clear whether we should actually be splitting them up, right? Primarily because it forms a good clade in itself. Whether taxonomy eventually will revise this to be 3 separate species, we're not sure. And again, it's not a complete sampling of the entire range. So if you can possibly think that if we have a good sampling in between the Red Sea and Great Barrier Reef and Okinawa, maybe those will fill in the gaps, right? Because now we are sampling from 3 disparate areas. That, of course, easy to think why they would form 3 separate clades. But maybe if we fill in the gaps, we might not have 3 distinct species. Sort of similar to the Hippolyta problem, Pachyceres speciosa, again, we all identify this quite easily. It's very abundant, one of the more abundant ones in Singapore, for sure. In Singapore, we find 2 different types, right? And globally, this 𝗵 and 𝗵 have shown that there are 5 different potential cryptic species, but definitely 3. So the red, the green, and the blue clades, right? And they separate, based on the genomic estimations, up to 10 million years ago. So 10 million years ago, they were starting to separate. So likely these will be 3 different species. The problem is it's very hard to morphologically identify them. They just look the same. So I think that's a classic case of cryptic speciation. But potentially, at least between the different regions, they can be distinguished. So there is at least only in Israel you find this particular group, and you don't find this particular lineage in Okinawa, for instance. So there are some clues as to how it can be revised, but currently no revisions yet. Because we have to again do the taxonomic homework, we have to find the nominal species and try to figure out if they have actually been described before. This has been changed. 𝗔𝘄𝗄𝗏, also fairly widespread. We've identified them as a monotypic genus, so 𝗔𝘄𝗄𝒏 𝗂𝘂. But Zoe Richards and her group have collected across Australia and parts of the Coral Triangle, as well as the Western Indian Ocean, and showed that there's a tropical clade and a temperate clade, and this is supported by mitogenomes as well as morphology. There's some distinguishing factors there. So I think those 𝗔𝘄𝗄𝒏 in Thailand is likely to be 𝗔𝘄𝗄𝒏 𝗂𝘂. So this is a junior synonym. Previously, junior synonym has been resurrected, right? So this is a big problem, because all you need to do is, if you are interested in the taxonomy, you just need to change the names, right? Because there's only one 𝗔𝘄𝗄𝒏 species in the tropical area and one 𝗔𝘄𝗄𝒏 species in the temperate zone. There's some overlap, but it doesn't really concern much of Southeast Asia. Okay, so again, if you want to use the right names, this is where to go, but it doesn't change our perspective of diversity. Okay, so I've described some of these revisions, these phylogenetic trees that eventually might lead to revisions. I'll describe a few challenges that may lay ahead of us. Even if we have all the data we currently have and we are trying to revise them, I think as you can see above, those are the majority of studies that have been done, and we are still lacking a good sense of most of the species in the region, right? So there are, again, there's a diversity problem, there's also a taxonomic bias problem, and then a biogeography problem. So I'll describe them a little bit as we go along. So there are over 1,600 species, right? 1,600 species. We have probably worked on less than 100 species so far genomically. So we are very, very, very far away from trying to resolve the entire tree. So that, in a sense, taxonomically, it is a problem, because if we are trying to resolve taxonomy, then we are only at the beginning, right? But it's only if we are interested in taxonomy, right? And in terms of global diversity, again, very high diversity for both zooxanthellae and azooxanthellae species. But if you look at the number of species that have been sequenced, at least for gene trees, most of them are zooxanthellae species, right? So this is a taxonomic bias that we need to be aware of. There are also certain groups that have not been sequenced very well, right? The groups, for instance, dendrophilids, right, associated with the zooxanthellates, both azooxanthellates and zooxanthellates, they have not been sequenced very well. A lot of rabbits as well needs a lot of work. A lot have been collected, but it is that transition from gene trees to genomics that caused a lot of problems, caused a lot of people to stop publishing, right? So I know of a few papers that were in the pipeline that were based on gene trees, and then the authors decided not to continue with it because the genomics are starting to come in, so they are now resequencing them for genomics, which overall may be a good thing, because you don't want to revise them with gene trees and realize that genomics is showing a different thing, and then have to revise them again. So I think that is not a bad thing, right? But I think we need to address the taxonomic bias in some of the collections that we have. Okay. Again, just to mention, right? So this is a relatively new study showing azooxanthellae species are also very diverse. In fact, a lot of new species that are discovered. Most of these new species have not been verified with genetics, right? Again, nobody's doubting them, but I think if we do more genetics on them, we might realize that there are some new lineages that might come in, and they might have to be studied in detail. All right, so I come to the last challenge, which is this, I call this the biogeography problem, which I have sort of alluded to before. If you have seen this paper by Tonbridge, right, and Andrew Baird, it's a very cheeky title called A Tenuous Relationship. And that's really just focused on one particular species, right? So we all go around identifying Acropora tenuis. We have it in Singapore, we have it in, or we at least identify in Singapore. I'm sure it's in Thailand. And it looks like this. Typically corymbos, right? Very even branches, evenly distributed branches. The radials are sort of cochlear-form and you know, lip-like, but also very even, sometimes forming this rosette. But that's kind of strange because if you look at the type, the holotype in the museum, it shows quite a different thing, right? It's not really, you know, it's not as even as it is, both in terms of the branch distributions as well as the radial distributions. The radials are in fact very uneven, very irregular. So, you know, Tonbridge and folks went around collecting from various parts of the Indo-Pacific, showing that actually what we have been calling tenuis actually can be multiple different species, and a lot of them have been revived from removed from synonymy from tenuis. So, for instance, Ksanthe is basically all of the species that all of the specimens that you call Acropora tenuis in Australia and Great Barrier Reef is actually Acropora Ksanthe, right? And so on and so forth. So you can see quite clearly though. So the numbers here, the first number in the specimen actually denotes the locality, and they are actually mostly from very restricted localities. When we look at Acropora tenuis, is even more so, right? Acropora tenuis is only found based on matching the holotype, found in Fiji and Tonga. These are the only two localities that Acropora tenuis is found. So I think the question we all have is, should we then call what we have in Southeast Asia Acropora tenuis if you see something like this? I think it sort of depends, because if you don't have a name for this, then you have to call it Acropora tenuis, but with a note, you know, we make sure we understand that this is actually probably not Acropora tenuis, probably a new species or other species, right? So this is one area where local identification still remains relevant. As long as you know we have this type, it's important then to figure out what this is. So collect it with the photos, skeleton, and DNA, match them up, and try to put that in the context of the global taxonomy, if that's a question that we want to address, right? But as for an ecologist, if it's a good species locally, whatever you call it, it's a good species, right? So this is what I call biogeography problem, because in order to resolve taxonomy, you now have to go to different parts of the reef, right? And it's really difficult to really collect, right? Because you go to one part of the reef, it's only one species. You go to another locality, it's another different species. Very difficult to collect for a comprehensive study, right? You need a lot of resources, a lot of travel money, of course, a lot of funding for sequencing genomes. Also, if you are interested in resolving the taxonomy, it's very hard to target species now, because we have been trained with the conventional taxonomy, and that is, if you go to a site, if it looks like this, it is this species. But in fact, it might actually be a very range-restricted species, right? It's more range-restricted than we expect, at least for Acropora, right? And so for Acropora, I think in terms of conservation, what this is telling us also is that there is actually a very high risk of extinction for many of these species, because they only are present in a couple of localities, right? So tenuis, Fiji, and Tonga. And so I think taxonomy has implications for conservation. This might be one where we need to start discussing what do we do, right? At least for Acroporas, because Acropora, again, is one of those indicative or keystone, what do you call it, this species where it's so signature for corals or for reefs. So what do we do with them, right? A lot of these range restricted species. Okay. But back to my, point is that taxonomy is global. This will be done as a global revision if we need to do it, but identification is local. So, and this re-emphasizes that because wherever you go, this species exists, but it may not exist as the same name that we have been using, right? So I think it's important to not just as a general statement that is local, but also that we need to recognize that the names we apply in one part of the world may not be applicable for another part of the world. So it's really important to separate them. And where we need to put them together, that's when we need to do the homework of matching photos to coral, skeleton to genes, and then putting them together in a taxonomic framework. Right, so come to the end, more or less the end of the, at least my content. So just some summary of what I've talked about. So this newer systematics suggests that many new taxa are still being discovered, especially 𝗔𝘀𝒧𝒪𝗃, a lot of novel lineages in 𝗔𝘀𝒧𝒪𝗃, expanding clade sizes. For 𝗂𝗏, it's definitely true. The clade sizes are going to expand because of these range-restricted species that they are finding. Now, the genomic data are only shifting the placements of those unstable taxa. So groups that we use gene trees to discover or to understand the phylogeny, if they are weak with gene trees, they may move around. So basal clade is one of those. In a way, basal clade is, is suggesting to us that it is something, that we don't really know what it is, right? So it's a basal clade. There's no distinguishing characters for this basal clade. So it tells us also that perhaps it is a good thing that the genomic data are showing that there is no such thing as a basal clade. Okay. And then finally, at the species level, that's where I think a lot of changes might take place. So we've seen the case for Leptestria, where you have a synonymy of Purinosa and Purpurea. You might see that for Porides in the next few years as more samples are being sequenced. You might see some of that definitely see that for 𝗂𝗏. Okay. Okay, so finally, I just want to maybe make a note that, I think we start out with this point that coral taxonomy is unstable. A lot of people think that it's confusing. I think we are not in a exceptionally bad time. I think I would acknowledge, of course, there are a lot of changes, but the changes haven't been really happening at breakneck speed. That is maybe a good thing, but it's also maybe a bad thing because now we have to contend with the fact that it keeps changing slowly a bit, changing slowly. But if you look at the past of coral taxonomy, right, since the Linnaeus, every few years, there has been a monograph, new species, new lineages, changes in the placements of certain species in general in families in suborders, and so on. So we are not really in an extraordinarily different time, right? So genetics really sort of started helping us in taxonomy in 2007 to today. Genomics started just a few years ago. And compared to the previous times, these are not, the changes really haven't happened at the speeds that we think has been happening. I think it's just because we are in it, so it feels like it is very confusing. But I would say it's even more confusing probably at the time of 1970s when Veron and Carden and Michel was putting this together in these different volumes, right? And they came out at every 2 or 3 years, one volume came out. I wouldn't think that would be an easier time. I'm sure they had to adjust. I'm sure some of you were in that time. Perhaps Nippon was in the time having to revise how we identify species at the time. I don't think we are in a very different time. I think the changes have been happening since 3 centuries. And I think that's the point about taxonomy, that it is a very different goal compared to at a local level when we are doing conservation. It may not be that important right? So I think as long as locally we are consistent with what you're doing, we can wait for to be updated on the taxonomy. It's not a big problem. Right. So with that, I thank you for your attention. I guess there's time for some questions, but I hope I will be able to learn. I'm sure I've learned from your questions. So happy to take more questions from you before you go for your lunch.
Hong Sensei, may I have question in Thai? Yes.
Yes, you can translate. Someone can translate for me, please. Thank you.
คือในช่วยถามแกว่าในขณะนี้ครับเทคนิคที่เรามีครับในเรื่องของคือเราสามารถจะรู้จีนที่เป็น resolute ยีนใน local species ได้หรือเปล่าตอนนี้ผมว่าไม่เข้าใจคำถามภาษาไทยผมแน่เลย yes เอาเลยพี่ชนช่วยหน่อยคือเราจะหา resident genes ใน species ที่เป็น local species เนี่ยเราจะพอรู้ได้ไหมครับผมขอบคุณครับ
The most updated technology we have in hands today, can we specify the gene that the gene that control the resilience of the coral? So he is more interested in resilience of the coral species. Can we identify that genes at the moment?
Yeah, thanks for the question. Am I muted? Let me check. Okay, so yeah, it's a question that I'm sure we're all interested in. I think there have been some studies that relate resilience. So in terms of bleaching resistance and also recoverability of corals, to specific genes that may or may not be present in some corals, and or the expression of those genes. So you may have the same gene, but it's expressed in higher rate in certain populations than the other. And I think that has been done. The problem is that there is probably no cause and effect that we can draw from this. So I think there are all association studies. A lot of them are from the genome by association studies. Some are from individual genes that are targeted for to look at transcriptomes or look at gene expression of certain genes. So we know these genes have differences under different conditions, and different species respond, different populations respond differently. But I don't think we know specifically whether they cause increased resilience or not. I think it's just still a relationship between certain genes with certain stresses, right? I think there is also this thought that that is not the only factor that drives resilience. There's going to be the symbionts as well. So symbiont types explains a very large variability of that, between whether a coral will bleach or not, or recover or not. Also the microbial communities. So I think there's a fair bit of all three. And that's why we consider the coral as a holobiont, right? So it's not just the host, but it's also the symbionts and also the microbes. Yeah. So this is not really my field, but I think based on my reading of it is that it's very complex, and a lot of people are now using the microbial communities as a way to enhance resilience. But also for the host is to sort of stress test them, right? So if you put them under sublethal conditions, they get better at adapting. So that sort of condition, a bit of thermal conditioning, and that might happen at the epigenetic level, and that is also something people are studying, whether within a single generation there are factors that are changing the way certain genes are being expressed. Likely it would be, but I think that's still something that needs to be done. Yeah.
So I'll translate your answer to the audience. So อันแรกคือสรุปว่า identify ได้แต่ว่ามันไม่ได้ consistency หรือไม่ได้สม่ำเสมอในทุกๆกลุ่มปะการังอันที่สองก็คือว่า gene expression มันยังไม่สามารถที่จะสรุปได้ชัดเจนว่ามันจะมีปัจจัยใดบ้างที่จะเป็นตัวควบคุมการแสดงออกของจีนที่ไปควบคุมลักษณะ resilience ที่เราสนใจอันที่สามก็คือว่านอกจากตัวปะการังแล้วนอกจาก gene expression แล้วยังมีอะไรประชาคมของแบคทีเรียแล้วก็ไอตัวซิมไบโอตอื่นๆที่ดำรงชีวิตอยู่ด้วยที่จะเป็นตัวควบคุมลักษณะการแสดงออกถึงความทนทานความสามารถในการเปลี่ยนแปลงตามสภาพแวดล้อมได้
Do we have any question else?
Thank you very much, Dr. Dan, for your 2 hours overview. This is very nice, very good. I just wondered if you have the form that writing in text about this talk that people in this room can go back and learn. Sometimes there's sometimes that they can get more understanding from the text.
Yeah, I think the recording there's a recording. I can of course share the slides. I'm happy to just send the slides to you guys. No problem. Yeah, I mean this is not my work. I mean I'm just putting, summarizing the really good work that people have done over the years, including folks in Thailand. I think it's fantastic. So yeah, I'll put this together and send it to you. But I think in terms of the text, I'm not sure I'll be able to translate it. But the recording, you'll probably be able to access it for sure.
Thank you.
Thank you.
Okay, thank you, Dr. Dan Wei. May I have got one question? It's quite simple question for the genetics, but it just I'm just wondering that how you can pick up just single one tissues from the whole colony and lab presence that is just only one sample of that's suitable for extract the DNA.
Yeah. Okay, so your question, I think maybe I'll interpret it is if you have a very large colony, which part is representative? For looking at genomics, well, looking at just barcoding or identifying species, any part of the colony is fine, right? But I think there is a global movement now to try to standardize the way you collect specimens. I think mainly coming from some Americans. I think they are trying to put data sets together, data sets of the symbionts as well as the bacterial communities. So again, of course, if you have a very, very large colony, there's some evidence to show that there's some somatic mutation. So if from a big Parades colony, you might get one genotype versus the other because of somatic mutations, but they're very, very minor. But in terms of, if let's say your study is going to be not just on identifying that species, but also looking at what symbionts they have and microbes they have, even epigenetics, then I think it will be, if it's very large colony, you have to take multiple samples, right? Multiple samples from the same colony, just to be sure you are looking at, the microbes or the symbionts that govern the health of the entire colony. But in terms of taxonomy, identifying species, doing genomics, phylogenomics, it is not a big issue to sample from anywhere. Of course, you would want to sample from where there's more tissue than skeleton. And that's for like, I mean, that's pretty easy for, for 𝗂𝗏𝗏 is just a branch. You are not gonna sample where the base of the skeleton, where base of the colony where it's actually more skeleton than tissue. So overall, I think it's mostly fine. Anyway, it's fine.
Okay, thank you. And if you don't mind, may I have another question next to this again? Because I'm quite interested in the case of the genomic DNA of the coral, as you just presented, because it's very impressive information, and this is a new skill and also new information for me as well. As I have just a little skill on DNA extraction from the fish, as the fish is just one single individual, that we can making sure that we can identify as the individual, and they extract the DNA out, and we can making sure that that is a represents the sample from the another geographic area that we want. But in terms of the coral, as some species, they are living as the colony, as you mentioned, and some of the species, and in some particular case, they also have sexual dimorphism, and also they also sometimes they don't they breed without sexual reproduction. Thank you. So that is make me wondering, and how we can separate that in terms of the genetic, and cause it might be one of the factors that interfere our information.
If I'm interpreting your question correctly, you're asking if different so whether male or female, or different parts of that, different generations of that same lineage can have a difference. Is that what you're asking? Whether you sample from different colonies, that they will be they have a different signature. Is that your question?
Yes, that is one of my question. That is the first question is in terms of the coral have like a sexual reproduction and sexual another asexual.
So her question is that are there any differences between coral that reproduce asexually and those species that reproduce sexually?
Asexually, basically. And so I think generally different species have different reproductive strategies. While we know 𝗂𝗏𝗏 𝗏𝗂𝗏 has both sexual and asexual. Well, they brood and then they spawn as well. So you have different modes of reproduction, and then there are also asexual versus sexual reproduction. I think generally no, because in terms of trying to understand their phylogeny, no. So basically, if you have new generations that are being produced, so if you have fragments versus if you have eggs or sperm, we are looking at the within species variation. If you are trying to understand between species variation, then there might be some differences. So let's say we are doing a population genetic study, right? So you're trying to understand how a species is structured. So whether one site is genetically differentiated from another site, then there might be some there are assumptions in those analyses you might do. So if you are doing parentage analysis, you may have to specify, for instance, are you taking from a direct descendant? So for instance, if you're taking an egg from a parent and you're analyzing those together, then you have to specify those parameters. But in terms of phylogenetics, if they are one generation apart or even multiple generations apart within the same species, they tend to be the same genomically. So we don't regard those variations as meaningful for phylogeny. But yes, they can be meaningful for population genetics. I hope that sort of answers your question.
Yeah, thank you very much.
Sure.
Anyone else, please? The next session is gonna be let's say it's gonna be thank you for Mr. Director of BMS.
Hello, Professor. Hello.
Hi.
Hi.
Is that for me? Yeah.
It's on behalf of the Institute of Marine Science, 𝗵𝗂 University, and the participants who attend this workshop, we would like to express our sincere thanks to Associate Professor Dan-Wai Huang, the Deputy Head, 𝗔𝘀𝘀𝒃 Natural History Museum, and the National University of Singapore. So from your presentation, we have learned a lot from your experiences and valuables of your presentation. Actually, we would love to invite you to have the presentation at our institute and meet our participants face to face. But we also have some kinds of difficulties to arrange and the limited anyway. So we would like to invite you again to our universities and make our collaboration between Thailand and Singapore and our two organizations. And we would love that you will be on your institute, and hopefully you can make a good collaboration with our participants, because this training program, we have many institutes from the universities, from the Department of Marine Environment and others, even the private sectors as well. So after this, I would like to ask our participants to give the approach to our speakers, please.
Thank you.
All right. So thank you very much, and welcome to Thailand when you have your free times, or even you have your vacations or others. Thank you very much.
Thank you. It's an honor. Thank you very much.
ครับผม ครับเชิญวิทยากรครับ
ค่ะสำหรับเซกชันนี้ก็การบรรยายภาคพิเศษนะคะจากหัวข้อนี้ก็สิ้นสุดลงแล้วนะคะขอบคุณดร.ห่วงเป็นอย่างมากเลยนะคะแล้วก็สำหรับการบรรยายภาคพิเศษนี้นะคะทางสถาบันวิทยาศาสตร์ทางทะเลได้มีการบันทึกวิดีโอไว้แล้วเดี๋ยวเราจะนำขึ้นแขวนบนหน้า Facebook ของสถาบันนะคะเนื่องจากการไลฟ์สดวันนี้ของเรามีปัญหานิดหน่อยนะคะท่านสามารถติดตามย้อนหลังได้นะคะผ่านทาง Facebook สถาบันค่ะแล้วก็ลำดับต่อไปนะคะก็ช่วงเวลาพักทานรับประทานอาหารกลางวันนะคะก็ขอเชิญผู้มีเกียรติทุกท่านนะคะผู้เข้าร่วมการอบรมทุกท่านรับประทานอาหารว่างร่วมกันได้ขอโทษค่ะรับอาหารเที่ยงนะคะพร้อมกันได้เลยนะคะตอนนี้เราจะมีพักกลางวันประมาณ1 ชั่วโมงนะคะแล้วก็จะมีบรรยายต่อในภาคบ่ายนะคะเรียนเชิญค่ะขอบคุณค่ะ Thanks, Dan.
Thanks. Bye.
Okay. Talk to you soon.
See you.
Bye.
จะชี้แจงเรื่องรูปภาพ